Okay, guys, it seems my slide did not get here on time. I'll talk about DECAAF. As you heard, it's delayed enhancement MRI determinant for catheter ablation for atrial fibrillation. We'll be presenting this today afternoon. So the purpose of the study is to use MRI to look at atrial tissue fibrosis as predictor of procedural outcome. So we did a multi-center study. The 14 centers were involved in the study between United States, Europe, and Australia. And every patient who was undergoing AFib ablation procedure had an MRI scan using specified optimized sequence to detect atrial disease or fibrosis. All these scans were downloaded to a blinded website. That means the operator who doing the procedure never knew what the atrium looked like in tissue, and were analyzed at the end of the study. Patients were followed up for at least 1 year. And we also looked at what the operators did in the study. Practically, we did MRI scans after the procedure at 3 months to 6 months after the procedure, looking at the delivery, the ablation, the type of lesions caused by the ablationist. As you all know, today in ablation of AFib, we're still debating what's the best strategy and where to ablate. And all the endpoints we have are electrograms endpoints. So now with this study, we're trying to see what really the operator do on the heart. It's the first study to describe in a multi-center fashion, not only how the atria or the hearts look like in tissue way before the procedure, but also what did the operator do for this heart? Where that the lesion sites were that impacted outcome. So we have 360 patients in DECAAF. We have 17% dropout rate in the study because of bad scans or other issues. We ended up with 260 patients, which we analyzed. The major finding in DECAAF is that hypertension was the only predictor or associated with fibrosis, with atrial disease in DECAAF. This is number one. Number two, fibrosis after collecting for 10 co-founders, including age, diabetes, hypertension, duration of AFib, participating center and experience of the center, fibrosis, the extent of atrial disease, was the only predictor for outcome. That's important. So when you look at an MRI, you can predict today what's the chance of patient having recurrence after ablation procedure, independent from the operator and experience center and the type of lesions. Another major finding in DECAAF, as you all know, sitting in this room, for the last 15 years, we've been targeting the pulmonary veins to isolate and treat AFib. That's a major finding as well, which we're sharing with today. The first time is that encircling the vein with lesions, you see them in MRI. So the MRI that we did after the procedure could tell me what did the operator did exactly. And these encircles of lesions were not important in terms of showing success, did not correlate with success. It means the pulmonary vein encircling of ablation lesions did not predict outcome. What predicted outcome is the fact that the fibrotic tissue, the diseased tissue, the more they were covered with the ablation region by the operator, we call it the residual fibrosis. So you have the initial disease. The more you cover of this with ablation, the better the outcome. I mean, there's something happening outside the atrium, which we all of us knew there's a disease going on, and there's microentent circuits and rotors and so on. But it seems that there's a lot happening outside the pulmonary veins, which we learned from DECAAF, again in a blinded fashion. So, and this applies, this rule, it seems from DECAAF, seems to apply more for patients have early stages of the disease, have little disease. If you cover the fibrosis, the chance of being cured is higher. It's 80% success if you have a little disease versus 30% success if you have a lot of scarring. So that's important. I'm open for questions.
Thank you so much. So we have time for questions. Please, gentlemen here.
Hi, I'm Michael Reardon with theheart.org. I was just, here. Based on your findings, do you recommend then against targeting the pulmonary veins? Like, how would this change, I guess, the technique?
Yes, I would target fibrosis, and that's what I'm doing in the lab, and that's it's encouraging, especially in early stages. If you don't find triggers, we still believe there's 6-8% of our population we're dealing with today that have triggers of AFib. But this is a big, big statement, as you can tell. Will be tomorrow everywhere in the world. Yes, I think we need to look at outside the veins more and more, and we're doing this. We're targeting fibrosis in every patient now to try to suppress the disease.
Does this reduce the time of the procedure? Like, does this shorten fluoroscopy time?
I mean,
what
that's
are the benefits of
I think the benefit is more important than the procedure time, and we're seeing it's the more you cover the fibrotic tissue, and now in this blinded fashion, the better the outcome. And the operators who were doing the procedure never knew what they have, right? They never knew the lesions they did. And it seems that if you put this lesion on top of fibrosis, you're improving the outcome dramatically.
There's this gentleman here, please.
Bruce Jansen with Cardiology News. So the ablation, I would think the ablation would also cause scarring.
Mm-hmm.
So you're
So
addressing the scar.
that's a very smart and very important question, and I'm glad you asked this question. The MRI, there's two ways of looking at. It's not every scar in MRI we're looking at the same. There's different thresholds. We're looking at fibrosis, which is a mix of still cells. If you look at the biopsy, there's a couple of papers published correlating this. If you look at the fibrosis itself, we see an MR. We're looking at fibrosis with healthy tissue in between, still viable. What the scar we're looking at in ablation scar, we call it the clean fibrosis, or clean, you don't see any cells that are left behind. That's what we're looking at in the MRI. So that's a clean ablation lesion. It's different from diseased tissue. It's completely different histology, and the threshold we use in MRI to detect both those. So it's two different things.
You have another question, please.
I do. I just a follow-up. So would you recommend that patients undergo an MRI prior to the, I mean, is that the only way to detect fibrosis?
That's the only way we have available to us today. And yes, I recommend everybody should look at their hearts before having an ablation procedure. And by the way, that's independent from age, independent from duration of AFib, independent from comorbidities, which will be shown more data today afternoon.
Any other questions? Please, in the back.
Is there also
question.
any, Mitchell Zola, Cardiology News, any link between the fibrosis profile and the disease severity, the presentation of the AFib?
That's another excellent question. So there's two patterns we're seeing. First of all, every single patient we saw in the study have a posterior wall involvement, every patient. And the more you see anterior wall involvement, the worse the news in terms of their outcome. Yes, so the more widespread fibrosis, the worse is the outcome.
So this is a new way to assess patients with AFib?
Patients with AFib is really predict outcome, and there's data published in the last couple of years relating this to stroke risks and so on.
Well, thank you so much. And now I hand over to Dr. Jessup, please.
Thank you. Our next study is entitled PROG 14, Perioperative Ischemia versus Perioperative